Today we're going to look at microbiology lab technique, which is culture media. So objectives for today's discussion are the types of culture media available in microbiology and difference between enrichment and selective medium. We're also going to look at the usage of both these mediums and the components of these mediums. We're also going to look at some tips for making a good culture medium. So as we go ahead, we have this question that what is a culture media? A culture media, which is also known as the growth media, is designed to support growth of microorganisms or cells via the process of cell proliferation. I like to think it of as food for bacterial cells. It can be present in three physical states: the solid, which is the agar plates; liquid, which is the nutrient broth in a test tube; and semi-solid. So there are many different types of media available in microbiology. I'm just going to list here a few: selective media, enrichment media, general purpose media, differential media, and supportive media. There are many more mediums available, but today we're just going to be talking about selective and enrichment medium. What is a selective medium? A selective medium is a culture medium that favors growth of selected microorganisms and inhibit the growth of other microorganisms. While an enrichment medium is a media supplemented by special nutrients, allowing growth of certain type of bacteria with specific or complicated nutritional requirements. Now let's look at the differences between enrichment and selective medium. Enrichment medium is present in liquid state, while selective medium is present in solid state. Enrichment medium isolates pathogen from a mixed culture, while selective media increases number of colonies of a desired bacteria. Enrichment media is basically adding nutrients for growth of a particular organisms. For example, controlling cultural conditions for a specific bacteria. Example, a bacteria grows better at a specific temperature or a pH, while selective media inhibits the growth of all other undesired bacteria. For example, adding antibiotic so only resistant bacteria can survive. So I look at it as sowing seeds and then only giving nutrition to one seed so it could grow better. So that's how I look at enrichment media. While, for example, selective media is giving poison to other trees so only one can survive. Now let's look at the components of these mediums. Different media are basically made for isolating particular organisms. They usually don't have the similar composition, but they should have some basic ingredients such as nutrient source, for example nitrogen and vitamin, energy source such as carbohydrates, essential metals and minerals, buffering agents for pH balance, selective agents if you're making a selective medium, for example antibiotic, and gelling agents, which is usually agar. There are different types of enrichment and selective medium. One of the examples of enrichment medium is F-selenite broth, which is used to isolate Salmonella and Shigella bacterial species. It is made of casein enzymic hydrolysate, which is a source of nitrogen and vitamin, lactose, which is a carbohydrate source, and sodium phosphate and sodium hydrogen selenite, which maintains pH. An example of selective medium is McConkey agar, which isolates gram-negative and enteric bacteria that particularly ferment lactose. It is made of peptone water, which is soluble protein source, agar-agar, which is a gelling agent, sodium taurocolate, which inhibits non-intestinal bacteria, lactose, which is a carbohydrate source, and neutral red, which gives it a distinctive red color. Let's look at how to make a good culture medium. A good culture medium should have a desired pH of neutral because a lot of acidity or alkalinity can kill the bacteria. A desired temperature for storage is 15 to 20 degrees Celsius before inoculation and should be stored away from sunlight because we don't want our medium to be invaded by surrounding bacteria. For isolating particular bacterial species, it is desired to use selective or enrichment media, while to investigate different microorganisms in a given specimen, it is desired to use a broad spectrum media.